Hello, welcome. We are in a new module now. We're actually moving away from the metabolic biochemistry related things that we've been on for the past couple of weeks. We're moving in a different direction. In fact, the way I see it, BIO110 has a little bit of a circuitous route. We spend some time talking about the fundamentals, then the biological molecules, basic chemistry underlying the biological molecules. And then we talk about cells for a while, then we talk about metabolic biochemistry, and now we're kind of coming back into that cellular focus once again. And this is a discussion of cell division, which is kind of like the beginning point for a discussion of things like genetics. And genetics will lead us into molecular genetics. Molecular genetics will pretty much be where we are for the entirety of the semester, for the final part of the semester. So yeah, we're making some pretty significant progress through the content of BIO110. Okay. Now, when it comes to cell division, this is a topic that a lot of students have had some exposure to. Not that you need to have had exposure to this in the past, right? But truth is that many students have come through some type of high school biology class, or maybe you've learned even about the different phases of mitosis. Okay, and so that's not one of the things that I tend to make a lot of put a lot of emphasis on, the descriptive aspects of what happens during prophase versus pro-metaphase. All that is totally fun stuff. I'm good with it for those. What I like to do is to get at bigger questions like, why do cells divide? Okay? And why do cells divide the way they do? Specifically, why do we have mitosis? And what is it about mitosis that allows cell division to take place in a nearly perfect way every time that it happens? Okay, and so these are kind of like the bigger questions that I'll be addressing with today's video, and so maybe it will be interesting even for those of you who have had some exposure to cell biology in the past. Okay. Now, the first thing that your book talks about, this would be in chapter 8, section 1. Why do cells divide? Right? And what is the whole point of cell division? Well, that's easy, right? For a multicellular organism like yourself, you had to grow from a small thing to a big adult, small child to big adult. And we know that growth does not occur through an increase in the size of the cells. The cells are always small, right? And if we have growth of an organism, it happens through an increasing number of cells. And hopefully your brain is clicking now and you're thinking about that argument that we put together a while ago, where we're thinking about like a surface area to volume ratio. Is that going off in your brain? And yeah, we're making lots of small cells, and that allows us to maintain the surface area required for the cells to do what they need to do, right? Another reason why we need cell division is because we have a lot of cells that don't last forever. And we have some cells that are pretty much final by the time you're born. Like, you hear that after you're born, you don't really have that many more nerve cells that come online. You don't really have that many more muscle cells that come online. But the majority of cells in your body have to be replaced. Right? Blood cells have a very short lifespan. Skin cells are continually replaced throughout life. And in order for us to have a continual supply of brand new skin cells and blood cells and liver cells, and pretty much most of the cells in your body have some need for replacement. Okay? That's going to require cell division. Okay? So yeah, check cell division. But the thing that your textbook notes is that if we look outside of the human orbit, there are plenty of organisms for which reproduction is going to occur through regular cell division. I'm talking about normal cell division here, not the kind that is associated with recombination of the genes, right? If you're thinking right now about having had a previous discussion, this is something I didn't really want to bring up, but there's always this thing about mitosis versus a different kind of cell division, meiosis. Okay. That discussion is something that I'm not even going to bring up today, but I'm mentioning it here because meiosis is the kind of cell division that's associated with recombination. And so as long as we're talking about the normal type of cell division that does not involve any recombination of the genes, the outcome of cell division is identical daughter cells. Two cells that are splitting off from the original cell. This is the cell originale, and then we undergo cell division. We end up with two daughter cells, daughter 1 and daughter 2. And there are no differences between the two daughter cells because we've had a perfect replication of everything that's there inside of the original cell. Okay, and this is significant for us because we're when we specify this normal type of cell division as being the mechanism of reproduction, the kinds of organisms that are being reproduced are going to be genetically identical to each other and also genetically identical to the original. In other words, this is a matter of cloning, right? Asexual reproduction is the same thing as cloning. Right? So if you have a bacterium and we've got two daughter bacteria, bacterium, right, bacterium. And then so these two daughter cells of the bacteria are identical to each other. You could say they're clones, right? And if you have like what is covered in the textbook in this chapter, let me go to the textbook right now. Okay, if you go to the textbook and see what they talk about, a yeast cell that is reproducing by cell division by asexual cell division by budding, the cell below, the cell that's down to the right, might be a little bit smaller than the cell that's up and to the left, but they're genetically the same. Okay? If you take a starfish and cut it into several pieces, if you take a starfish and cut it into 7 pieces, each arm, if you cut each arm off, each arm will regenerate into a full-on starfish. And when that happens, the starfish you're ending up with is identical to the starfish you started out with, right? And so, yeah, I mean, if you did that, you cut the starfish up into 100 different pieces, and each piece grew into its own starfish, that would be kind of a matter of cloning. And we use this not because we're always that interested in propagating starfish, but when we have, we do the same thing with plants. If you take a fig tree, if you have a really good fig tree, you could cut the branches off, cut little sections of the branch, and for every section, you could have it sprout roots. Each little branch section will grow into its own fig tree, and all those trees would be identical to each other and identical to the parent fig plant, the mother plant from which you're cloning. And so, yeah, these would all be examples of asexual reproduction because they don't involve any type of recombination of the genes, right? And so, so just to point out that, yeah, there is the other kind of cell division that we ultimately are going to care about, but not yet, right? The other kind of cell division is going to involve some type of recombination. And that's going to be something that we'll be bringing up shortly before going into the phenomenon of genetics. Okay. So that's 8.1. That's kind of like a summary why we're reading 8.1. 8.2 is a really quick read. And the idea is that, well, if we're talking about prokaryotes, and here really thinking about bacteria, right? Bacteria do not undergo mitosis. Mitosis is a really sophisticated phenomenon. It's limited to eukaryotic cells. And it's a real fancy thing where the chromosomes are doing fancy stuff. In prokaryotes, the cell is small enough, genome is small enough, where all you have in the genome, if this is a bacterium, all you have is a big, in the nucleoid region, you have a big circle of DNA. And when the circle is divided, when you've got two identical circles, then the bacterium can split into two identical daughter cells that will fraction off from each other. And each daughter cell is going to be receiving the same gigantic circle of DNA. In other words, the phenomenon of cell division in bacteria is very simple. We say it's binary because one is splitting into two. Fission is the word that means splitting, okay? And that's kind of like the starting point. We know that eukaryotic cells also undergo binary splitting. One cell splits into two, although the actual process that's involved, is in particular leading up to the actual splitting of the two cells, is complex. There's this well-described song and dance, we call it mitosis, where the chromosomes thicken up, they line up at the metaphase plane, they separate, and eventually we have the formation of new nuclei. And well, that is the song and dance that is undergone when the cell happens to go through mitosis. What I want to do at this point is to look at, I think it's 8.3, the eukaryotic genome structure. We're really understanding why it is that mitosis is so important. And it really comes down to this, right? The eukaryotic genome is really huge compared to a bacterial genome. All the DNA of a bacterium might seem like a gigantic molecule, but it's not nearly the size of what you have in a typical eukaryote. And for me, understanding why it is that the eukaryotic genome is so much different from the bacterial genome has to deal with our need to organize all of that material so that when we have the splitting of the cells, when that one cell splits into two, we do actually get a perfect replication of the genome. Right? We can't afford for there to be any mistakes or some genes to be copied imperfectly between the daughter cells, because that will only lead to further mistakes in the future. Right? Once those two daughter cells split apart, if one gets the wrong genetic information, it's not like that daughter cell is going to be able to repair that mistake. That's going to be the genome that it has forever going into the future, right? And so because of the high stakes that are involved with the replication of the genome, because of the size of the genome, we have to have this characteristic structure. And this is the part of 8.3 that a lot of people have some difficulty with, okay? And so we have you can call them the chroma words, 3 words that seem really similar: chromatin, chromosomes, and sister chromatids. Okay? And I'm going to talk you through them on a different page, and we'll also look at what the textbook has to say about them as well, right? And so the genome structure of eukaryotes, okay, is different from the bacteria. Remember, bacteria have a big circle of DNA. Genome structure of eukaryotes occurs in many lines, right? So it's supposed to be a contiguous line, many lines of DNA. Okay, because some are shorter, some are longer. Our cells usually have them paired up, right? So these might be 6 chromosomes, and the information that's on the chromosomes, the sequence of A's, C's, T's, and G's is going to be the information that's carried in the genome, right? And so far, you're good with that. Okay? Now we know that there's DNA, okay? But we're also going to have another material here. Okay, it's a protein, and the main protein that we're talking about is a protein called histone. Okay, and histone proteins are like little beads, like little spools. And what happens if you were to look at this histone protein, kind of looks like this, right? And what happens is the DNA is a double-stranded molecule. It is a double-stranded molecule. I'm not going to draw it as a double-stranded molecule, but what happens is the DNA coils around the histone, 6 turns, then it comes over and coils around the histone. It goes and coils around another histone, which I haven't drawn, right? And what the histone does is it allows the DNA to be carried in an organized way. It's basically kind of like uh, putting all the papers into file folders, right? And by having the histone protein, we're able to manage a huge amount of DNA much more effectively than we could if the DNA were just sitting there naked and stretched out and subject to all the bad things that could happen to DNA inside of a nucleus, right? And so this combination of DNA plus histone is the material that we call chromatin. Okay? When we talk about the DNA sequence, we're only thinking about the DNA, but in an actual cell, that DNA, that long molecule is complex with histone protein into this material called chromatin, which is the stuff that we actually see inside the nucleus. This is the stuff we see as chromosomes. Okay, and so I guess the idea here is that the overall genome is organized into chromosomes. Linear fragments of DNA going from one end to the other, from one end to the other. And so this would actually be 6 chromosomes that we would have in the nucleus of this cell. And I've stretched them out so you could see them. And then to understand what is actually the material that we see forming during mitosis inside the nucleus of our eukaryotic cell, that material is chromatin, which is this combination of the actual DNA, the DNA portion of the chromosome, together with the histone protein that allows a long molecule of DNA to be compacted and visualized as a material that is much shorter. There's the actual length of the DNA might be several centimeters, okay? But there's actually only a few millimeters worth of chromatin in each chromosome when this thing is completely compacted. Okay, so we get some level of compacting as the DNA in the chromosome is combined with the histone protein. Okay. Now the other word here has to deal with the sister chromatids, okay? And the reason for that is that we know that in order for us to undergo cell division, let me erase these. Come back down. We're just going to visualize these full-on chromosomes. In order for us to actually undergo cell division, we know that we need to have 2 copies of the information on every chromosome. So this chromosome has got to be doubled. Oops, it's supposed to be different color, so let me undo that. Draw purple. So at some point, we have to take all the information in the original DNA strand and make a copy. Right? It doesn't happen exactly that way, but for now, what we want to think is, well, if we're going to be separating all of these chromosomes into 2 daughter cells, we need to have 2 copies of each chromosome. Okay, in which we've got 2 identical copies of the same genetic material. Okay? And so when this happens, the 2 things that we have here are called sister chromatids. And these sister chromatids are held together by a structure that we'll identify as the centromere. And so then that might be the centromere for this pair of chromosomes. This might be the centromere for this pair of chromosomes. Maybe this one's upside down relative to the other. This might be the centromere. It doesn't have to be in the middle. It could be at the very end. This thing, it might be the centromere for this third pair of chromosomes, right? And then, yeah, this has to happen because in the process of cell division, we have to end up with two daughter cells. Each daughter cell is going to have one of each of these, one sister from each of these pairs of chromatids, right? And so what some people have a little bit of confusion about is that at this point, after we have the replication of DNA, in other words, after we undergo DNA replication, so that we've got two sister chromatids, okay? This unit that I've just outlined in red, this corresponds to one chromosome. That has been doubled because it's ready to undergo cell division. And that chromosome consists of two sister chromatids. Okay? And so, again, yeah, there's a lot that goes on here, okay? But we have to have our minds pretty clearly around why it is that chromosomes are sometimes depicted like this, like little X's, and sometimes chromosomes are also accurately depicted like this, where you might just have a centromere and we don't have them doubled, right? They're single chromosomes, one chromosome, one chromatid. They're single chromosomes up until we undergo DNA replication, right? Okay. After DNA replication, each chromosome is comprised of two sister chromatids, right? And now we can go to the textbook and see what how it is described, this same phenomenon. Again, I like to do this because my impression is that when we come to certain junctures in the content of a biology class, like with three words that are kind of similar, like chromatin, chromosomes, and chromatids, we have a tendency to kind of gloss over and not pay attention to what's going on. And so, yeah, this is basically exactly what we talked about, right? If you scroll through the same chapter now, and if it wasn't making sense to you before, maybe things are better now, right? So here's the thing, okay? So here, this on the top would be a chromosome. My annotation function. This would be a chromosome before replication, okay? This would be the chromosome after replication. And the centromere that we drew before is going to be there, right? Now, during mitosis, these two chromatids are going to split apart, and one chromatid is going to go into each daughter cell. And the whole reason for doing this, I could understand this as being simply a very organized way to make sure that the two daughter cells are going to very reliably get exactly the same genetic information. Okay. Now, and by the way, this thing here is a chromosome that has undergone a lot more packing. This would be a chromosome at metaphase, which means it's already highly, highly compacted relative to the way that the chromatin usually exists, right? So remember I said that, well, DNA is wound on histone proteins, and so it kind of looks like little beads on a string. That would be chromatin in its normal state, and then this is going to undergo further packing, so the beads on the string are going to coil, and then the coil is going to coil again. And after a couple of rounds of coiling, it starts off as something that is very, very thin, becomes much thicker and shorter chromatid. And that's what we're looking at here. These sister chromatids, the sister chromatid right here, it's actually one contiguous piece of DNA, lots of histone proteins that starts off impossibly thin, but then it undergoes a lot of packing this is during prophase, where the chromatin is becoming thicker and shorter, such that by the time we get to see it at metaphase, it'll be this visible entity that we can actually watch separate and go into the two daughter cells. All right. And again, this discussion should make a lot of sense, presuming that you've already been through the reading, you've had a little chance to read through, maybe struggle a little bit, maybe not, okay? But at this point, we are going to skip over into the next bit, which is going to be the cell cycle. Okay? I'm going to stick with the textbook and bring up this diagram here, which describes the cell cycle. Okay, and I'll tell you exactly what's written on the whiteboard. We'll go back to the whiteboard really shortly. Okay, we said the cell cycle has two main parts. We have interphase, and then we've got mitosis, right? Mitotic phase. And so cell cycle equals interphase plus mitosis, if you include cytokinesis in there. Okay? They could call it the mitotic phase. In the textbook, I just think M is the mitosis part of the whole process. Now, mitosis is the part, this little section of the cell cycle, right? Looks like it's only about, maybe a 20th of the overall cell cycle. The mitosis part is the part that's divided into prophase, prometaphase, metaphase, anaphase, and telophase. And so if you look at that part, that's where you have the prophase. Sometimes we have a prometaphase, I think your book does. Let's just say metaphase, because I'm not going to be that interested in the details here. Anaphase, and then telophase. Somebody somewhere is going to ask you to know the different phases of mitosis. I can promise it won't be in my class. Now, the thing that I love about this diagram is it accurately depicts the fact that the majority of the cell's existence is not in mitosis. The majority of the cell's existence is in the interphase, the time in between cell divisions, right? And so it also says that we've got interphase consisting of G1, which is a gap phase, S, which is a synthesis phase, and then we have a second gap phase, G2, right? G1, S, G2, and the S, standing for synthesis, is telling us very clearly that this is the part in which we have DNA synthesis, right? At the very beginning, the chromosomes look like this, right? Each chromosome has one chromatid, right? One chromosome, one chromatid. And in the process of undergoing DNA replication, by the time we get to the end of the S phase, that chromosome is going to consist of two chromatids that are joined together at the centromere, right? And so, yeah, the life of the cell consists of a long period in which we're doing some growth. This is where the cell is actively doing what the cell needs to do. That's G1. The cell is doing more of that, only with an extra copy of the chromosomes. That's happening with the second phase. And through some significant portion of that middle portion of interphase, we have DNA synthesis, right? In other words, during the synthesis phase, we're not really seeing a whole lot of other gene activity apart from the replicate, because all the genes are in the process of being replicated. Okay, and so yeah, this is an important thing for us. 8.5 is where the textbook will get into a lot of detailed description of the different phases of mitosis. It's something that I have always thought was kind of pointless. Okay, maybe not completely pointless, but it seems silly to spend a whole lot of time getting into descriptive detail of some thing that happens. It's a relatively minor part of the actual biology of a cell, right? But let's look at what's going on there. Okay, so this is kind of what people expect to see when you hear that the lesson is going to be about cell division. Oh yeah, we're going to be learning about the stages of mitosis. You've got prophase, prometaphase, and tells you what's going on. You've got metaphase happening. This is actually pretty cool because it shows you the spindle forming little bioflicks. You can feel free to watch that if you like. And so you can speed through it. It tells you what's going on. I'm not gonna make a big deal about that. Then we have the rest of mitosis, metaphase, anaphase, telophase. Cytokinesis is really the part where the two cells are splitting, right? Anaphase is when the two nuclei, telophase is when the two nuclei are beginning to form. When you actually have the two cells splitting apart, that cytokinesis in your book actually goes through a whole section of chapter 8, talking about the differences between cytokinesis for animal cells versus plant cells. I'm having you skip that, right? The next part that is of interest to us, though, okay, has to deal with our first opportunity to talk about how the cell cycle is controlled, right? In fact, the way I see it, the biologists that are really interested in mitosis are cancer biologists, right? Why are cancer biologists interested in mitosis? Well, cell division, right, is kind of a characteristic of cancer cells. Cancer cells in particular are not doing something, are doing things that are not particularly well behaved. It's kind of like we've got a normal set of behavior of cells, and when the cells start misbehaving, that's basically what constitutes a transition from normal behavior to the behavior of cancer cells, right? And so this chapter 8, section 7 gives us a really nice picture of what cells are kind of supposed to do, okay? And even though this is giving us a picture of cells that are growing inside of a tissue culture, the same kind of thing happens along a base membrane in your kidney or your skin, right? This is what happens normally, and it's really kind of beautiful, right? It's like this. We know that we rely on cell divisions to grow or to replace damaged tissue, so we need to have cell divisions up until the point where we have all the cells occupying the spaces that the cells have to occupy. Okay? And once all the cells have filled in the gaps where they need to fill, we need to have those cells stop dividing. Okay? So this normal behavior of, you'd call it density-dependent inhibition, is illustrated here because once the cells run out of free space on the surface of the tissue culture dish, they stop dividing, and that's good. That's beautiful. That's exactly what's going to happen, right? And if you take some cells away, the cells are going to be starting to grow again, and we basically fill all the space that needs to be filled, and then once they're in contact with each other, they stop growing, right? That's normal behavior. If you take away that normal behavior and prevent the cells from ever stopping their division, in other words, we're supposed to stop the cell cycle, but if we continue to have cell cycles in more and more cell divisions, that is what constitutes cancerous behavior. And a tumor is basically a big lump of cells that was supposed to stop dividing a long time ago, but they're continuing to divide, and we are getting a little bit concerned about this excessive behavior because those tumor cells, all those divisions are going to cause disruption in your normal physiology and normal anatomy. Okay. And so that's really good illustration of why we're interested in things like cell divisions. Far more interesting. This basically sets us up with a much better motivation compared to simply learning about different stages of mitosis. The other illustration here about the way that that the cell cycle is controlled is through kind of extrinsic factors. Okay, and so in other words, in the case of density-dependent growth, the cells themselves are controlling their own growth, right? And in other cases, we have cells that require extrinsic factors in order to initiate the growth process. Right? Cells can respond to outside signals to initiate growth, and that's what's basically being demonstrated here. We don't simply have cells that continue to grow. They're supposed to stop growing after a certain point. In a lot of cases, the cell growth isn't going to happen unless we provide it with the appropriate conditions of nutrients as well as the chemical signals that allow them to continue to grow. All right, time to finish up here. I'm going to go back to my whiteboard, go back to my original outline, and make sure that we've covered everything that we need to cover, right? This part I think is really important, right? Understand why it is that we have this big discussion of chromatin and chromosomes and sister chromatids, why this organization is important. Well, we need to have this perfect cell division, right? Your cells are undergoing dozens, hundreds of cell divisions over the course of your lifespan, maybe even more than that. Every time you have to come up with exactly the same genome. Because if you don't, that's actually one of the things that contributes to the development of cancer. Okay. The cell cycle is a larger context within which the cell divisions of eukaryotic cells occurs. Interphase and mitosis are the two parts of the cell cycle, and cytokinesis is when the two cells divide. The last thing that we identified, we illustrated using density-dependent growth and the importance of growth factors, at least for some populations of cells. It reminds us that the normal behavior of cells, the cell cycle, is highly regulated and controlled. Okay, that's it. I'll see you next time. We'll talk about more fun stuff relating to cell biology.